Yes, so I would like very much to thank Radha and Joyce for organizing such a wonderful meeting. It's been a lot of fun, and I've learned a lot, and I've enjoyed meeting many new friends and old friends. So the question I want to talk about today is how DNA gets packaged into mitotic chromosomes so that it can segregate to daughter cells. And I'm going to show you an example of this in a fabulous movie made by John Dahm in Gary Gorpsky's lab. So here you see a cell nucleus. And if you just watch it, you see what's first a subtle change and then a dramatic change as the DNA reorganizes itself into mitotic chromosomes. Absolutely fascinating problem. Now, mitosis is a remarkable structural transition. Mitotic chromosomes were actually first observed in the 1840s, but they were really first described well by Fleming. And so this question is 136 years old. How do those chromosomes form? And what is their, and actually to understand how they form, we need to understand what their structure is. So let's put the problem in perspective. The human haploid genome is 1.2 meters. So in each cell there's 2.4 meters of DNA, okay. The diameter of a typical cell nucleus is 6 microns. Let's make an analogy. If a cell nucleus was the size of a tennis ball, the length of chromosome 1 alone, just one chromosome, would be longer than 40 tennis courts. So how did we accomplish this 10,000-fold compaction to get that amount of DNA packaged into that mitotic chromosome so that it can segregate? Now, work from the Uli Lemley's lab done before I joined it and while I was a member of the lab showed that mitotic chromosomes are organized into loops. And here you can see a loop, and on the right you see the nucleosomes on that loop. Okay. What holds the loops? What's at the bottom of the loops? Well, Uli proposed that there's a protein scaffold along the axis of each sister chromatid. And he proposed that that scaffold was made of non-histone proteins, which at the time he proposed it were completely unidentified. But then there was a huge fight, and this was labeled an artifact, and for many years you couldn't publish a paper on this subject, and you certainly couldn't get funded to study this subject if you mentioned that there was a scaffold in chromosomes, because it was an artifact. So when I set up my own lab, and in the years since then, one of the questions that we wanted to ask is just what are mitotic chromosomes made of? And the work where we got our biggest answer from that was using methods that I'd learned in Uli Lemley's lab. We isolated the chromosomes, and then we characterized them first by gel electrophoresis and making antibodies, and later by mass spectrometry. And by mass spectrometry, we could identify over 4,000 proteins in chromosomes, and that was in 2010. If we did it today, we would probably get 6,000 or 8,000 proteins. I mean, there's a huge number of proteins in mitotic chromosomes, most of which are actually not that important for the structure of the chromosome. But there are key proteins in one of the red quadrants here that are extremely important for chromosomes. And this is a protein complex, of which there actually are 2 in most cell types, called condensin, which was named by Tatsuya Hirano and actually identified in a number of different laboratories at the same time. Different subunits of the condensin complex were identified, but Tatsuya Hirano gave it its name. The trouble with condensin was that after it was identified, it remained controversial for many years exactly what it did. So in paper that is currently in press in the Journal of Cell Science, we used an auxin-inducible degron to get rid of condensin from cells extremely rapidly. So with this degron system, when this takes advantage of, so those of you who work on animal cells may not know that the way that plants control the response of genes that are responsive to auxin is that there's an inhibitor sitting on the genes, and when you add auxin, the inhibitor gets degraded, and then the genes are activated. So animal cells don't care about auxin, so you can use the target sequence for the auxin and the F-box and its target F-box protein, and you can inactivate proteins extremely rapidly. And when you do that, you see something like what you see in this electron micrograph here. So this is an entire mitotic cell sequenced by serial block-face SEM and using a Gatan 3-view microscope done by Dan Booth. He's now modeled the chromatin, he's identified the chromatin, and the chromatin is now labeled in red. And now he will segment individual chromosomes in that mess. So that doesn't look like mitotic chromosomes at all, it looks like a mess. In fact, I think it looks, to me, it looks sort of like a brain. It doesn't look like chromosomes at all. But you can see the different colors tell you that there are different chromosomes in there. And so, it turns out that if you now analyze that, In wild-type cells, you can easily recognize chromosome 1. It's the largest chromosome in the cells. In the condensed and knockout cells, I'm not sure that we can recognize chromosome 1, but those two structures that you see in the bottom right, which just look like lumpy messes, are the two largest structures in that cell, so those are probably or possibly chromosome 1. And so you can see, in the absence of condensin, if you get rid of it extremely rapidly, the chromosomes look extremely disorganized. But interestingly enough, the volume of those chromosomes is the same as the volume of the wild-type chromosomes. So the compaction of chromatin is not what condensin does, it does something else. So conclusions thus far. There was a 20-year controversy, but after the result of that was non-histone proteins do indeed have an important role in mitotic chromosome structure. Condensin complexes have a key role. Condensin does not simply compact the chromatin, it does something else to regulate the overall architecture. And that's what we would like to understand. So, in recent years, there's been a breakthrough in studying nuclear organization, which has turned into a breakthrough in studying the structure of mitotic chromosomes as well. And this is a methodology developed by Job Dekker called Hi-C. He developed a series of technologies, the C technologies, all of which are involved in determining which proteins, which DNA sequences are near one another in the nucleus. And the way you do Hi-C is you take a cell, you fix the cell with formaldehyde, and that fixation will trap pieces of DNA that are near one another just because it cross-links protein and the DNA is trapped in the cross-linked protein. And now those could be DNA sequences that are right next to each other along the chromosome, but they could be even two separate chromosomes that just happen to be next to one another. So what you do is you label the ends with biotin, ligate the DNA molecules together, break off the ends, and sequence the junction between these fragments of DNA that were trapped by a common protein. And in the bottom right, you see the image of an interphase cell, and these squares that you see are called TADs, or topologically associating domains. And that structure you're seeing is actually a heat map, where red means that you saw lots of crosslinks between adjacent DNA molecules. And as you can see from the blue arrows just to the right of it, if you have a spot that's off of that axis, for example, if you have, and you plot the same chromosome across the top and the same chromosome down on the side, that's what gives you the diagonal. You can see that region 10 of the chromosome is cross-linking to region 7. And the only way you can explain that is if there's a loop. So this was this is really, has a very high impact on studies of nuclear organization. But back in 2013, Job Dekker and his collaborator Leonid Mirny decided to look at what happened when cells entered mitosis. And what they saw, instead of this organization of TADs, and then the sort of Scottish plaid that you see off of the diagonal reflects longer range interactions, and those are called compartments. Instead of TADs and compartments, in mitotic chromosomes, you just saw the diagonal and a sort of haze that surrounded it. And the best interpretation of that haze, it's complicated, and if you want to talk about it, come and talk to me afterwards, we can talk about it. But the best interpretation of that haze is that chromosomes are organized as loops. And the loops are random, they're not sequence determined, they can be anywhere along the chromosome, and the loops are about 80 to 120 kilobases in size. So that's what they deduced. And I was excited by this. I was at a meeting where they presented this work before publication. And that was actually the same size of loops that I measured by doing electron microscopy of chromosomes. Back in 1981, I measured that there were 80 kilobase loops. But it seemed to me that we could get a lot further than the information that they had. So what we wanted to do was we wanted to define the pathway for mitotic chromosome formation. And the result of conversations that began at that meeting were a collaboration between my lab in Edinburgh, Job Dekker's lab at the University of Massachusetts, and Leonid Mirny's lab at MIT. And this is the perfect collaboration. My lab did all of the biology. Job Dekker's lab did all of the Hi-C and all of the complicated analysis of the Hi-C data. And Leonid Mirny's lab are physicists, and they did all the mathematical modeling that was required to understand this. And this work is just now in Press and Science, if you want to have a look at it. So what we decided to do was that in order to see the pathway of chromosome formation, we needed to have all the cells enter mitosis at exactly the same time. And how could we do that? we could take advantage of technology developed by Kevin Shokot in chemical biology to make an analog-sensitive allele of the kinase that John Pines was just talking to you about, one of the proteins he was talking to you about, CDK1. It turns out that if you inhibit CDK1, you will not enter mitosis. So the Shokot technology is to make a mutation in the ATP binding pocket that makes it a little bit bigger, and you can get an artificial substrate shown in the lower right, 1-NMPP1, and the 1-NMPP1 was synthesized for us by my friend Jim Paulson in Wisconsin. And that 1-NMPP1 will inhibit CDK1. And when it inhibits CDK1, the cell will not enter mitosis. Now there's another very nice property of 1-NMPP1, which is it's a very well-behaved molecule. So if I have a culture of cells that are inhibited by 1-NMPP1 and I wash the cells, the 1-NMPP1 will wash right out quickly. And then I can reverse the inhibition of a CDK1. And I want to show you what happens. And this is what Kumiko Samajima did. So here is a culture of chicken DT40 cells inhibited with 1-NMPP1. And they actually all look quite similar. And in fact, they're blocked at a point very late in the G2 phase. And if she washes out the 1-NMPP1, that's 0 minutes. By 5 minutes, all of the cells are in prophase. By 15 minutes, they're all in prometaphase. And by 30 minutes, they're in late prometaphase and heading into metaphase. And by 60 minutes, they're all in metaphase. I mean, we're talking, for a cell biologist, this was just mind-boggling. 96% of the cells are in prophase. To get that sort of synchrony was just was quite unexpected. So what Kumiko did was she prepared samples from these cells in Scotland, then sent the samples to Johan Gibkus, who's a senior postdoctoral associate with Job Decker. And it took a while. You know, these collaborations all take a little while. One lab sends the samples, the other lab's kind of busy. They don't do very much, but eventually they try an experiment. And I think I was on my summer holiday when I got this email, "Oh my God, what's going on? I can't believe this." And that was from Johan, and they had never seen anything like this. So this is the high C profile, like we saw before, of an interphase nucleus. Now let's look at a high C profile of a culture where every cell is in the same phase of the cell cycle, but it's still interphase. And what you see is that profile becomes hyper sharp. So that actually tells me that there is a preferred organization, at least when you look at the nucleus in this way. And remember, the diagonal is looking at interactions close to one another in the chromosomes, and the plaid on either side is looking at long-range interactions. So now we're going to wash out the 1-NMPP1, and within 5 minutes you see a dramatic change. The long-range interactions just melt away. By 10 minutes, the long-range interactions are gone. There are no long-range interactions. There's still some short-range interactions. But by 15 minutes, those are gone completely. And now this is the oh my God moment. Something happens. The red bar on either side of that line looks different. And if we look a little bit later, we see two diagonals appear. What is going on there? So in a high C map like this, when you see a diagonal like that, the distance away from the diagonal reflects how far apart you are on the chromosome. And when you see a diagonal like that, which is located at a distance of 2 to 5 megabases away in this map, that says that every sequence in the chromosome is now interacting with another sequence that's 2 to 5 megabases away. And then it's a little further away. Well, and you see, so that spreads out and gets further and further apart. So first they're interacting with sequences 2 megabases away, then 5 megabases away, then 10 megabases away. What's going on? It turns out that there are 2 transitions in metaphase chromosome formation. The first step is formation of loop arrays during prophase. And this looks like a very uninformative plot to you, I'm sure. And it was very uninformative to me at the beginning. What we're plotting on the y-axis is the frequency with which 2 sequences interact with one another, and what we're plotting on the x-axis is how far apart they are. And what you can see is that sequences close to one another interact more, and as they get a little bit further apart, they get a little bit less likely to interact, but then at some point they become much less likely to interact. That defines the size of the loops. And the model that works to explain this is that the chromosomes are organized in loops which are 60 kilobases in size. But now what's going on with this second diagonal? So we had the chromosomes interacting with sequences all along the loops, but now we have this second diagonal appearing, spreading and moving out, and you can see it in this probability plot as a little bump that moves out like that. The only way you can explain this is if the chromosome is a helix. So that in addition to interacting with sequences nearby itself, it interacts up the length of the helix. And in the beginning, the helix is long and thinner, so the sequences above one another are 3 megabases apart, but the helix sort of screws itself around together and becomes shorter and fatter, so that later on, those sequences up the way are 10 megabases apart. And to fit this data, what we propose is that the chromosome must be organized by an axial scaffold, which looks like a spiral staircase. And what you can imagine is coming off of the end of each step of the staircase is a loop coming out. And there's this loop, and then the next loop is one step up and over and comes out that way, and the next loop. But the loops are not perfectly ordered. And to test that model, Anton Golub-Dorodko in Leonid Mirny's lab did a mathematical simulation, and that's what you see on the right. And you can see the two images that you look on the right look very, very similar to one another. And the plot, the contact probability plot on the bottom right, you can see the red and the blue curves are superimposed. So that model really explains what's going on. And this can explain how you can have a combination of a chromosome which is a helix. It has loops, but as you see on the left, usually when you look at chromosomes, you don't see anything that looks either like loops or a helix. So how do we explain all this? So coiling of mitotic chromosomes has been known since 1880, but it's really difficult or impossible to see that on native chromosomes. So Kamiko Samajima has been doing super resolution microscopy in my lab and trying to see if she could get better imaging of some sort of coiled scaffold within the chromosomes, because we could deduce from the high C data that there must be a helix. And in fact, it turns out it's very hard to see, but if you look at the chromatid on the top left, you can see a squiggly, curly little thing. And what that is that's one of the components of the condensin complex, which has had GFP knocked into its endogenous gene. And indeed, it is making a sort of a helical axis on the chromosome. So it was possible in some cases to see that, although it's not easy to see it. But then we did this modeling, and we had a huge problem. And the problem was we could predict the size of the loops. We knew how much DNA was in the chromosomes, and also because of the serial block face SEM that we'd been doing and measurements that Kamiko had been making, we knew what the volume of a chromosome was. And the trouble was, you couldn't put the clothes into the suitcase. You couldn't fit all the DNA into the suitcase. So what was going on? And it turned out that got solved by the mathematical modeling of Anton Golub-Dorodko. And what he deduced was that there actually there are big loops, and then the big loops are divided into smaller loops inside. Now you can't really see that here, but if we look at this picture where it's the same image, you can see 3 different colors. And you can see in the middle some blue rings. The blue rings are a molecule called condensin 1. And condensin 1, well, the red rings is what makes the helix, and that's a molecule called condensin 2. So condensin 2 makes the helix, that's the scaffold. Condensin 1, the blue loops, associates with the scaffold, but it makes loops within the other loops. And I'll just very quickly show you how this works. There's 5 times more condensin 1 than there is condensin 2, and condensin 1 comes on and off the chromosome really quickly, but condensin 2 stays. So if you imagine that each condensin ring has a loop of DNA that it traps a loop, and the loop sort of grows out of it and gets bigger with time. It's called loop extrusion. This is what that will look like. So there's condensin 2 making big stable loops, but now condensin 1 goes on and it's flickering on and off. And the only way you can explain this is if it makes loops within those bigger loops. Okay? So now let's go back to the question of those condensin molecules and chromosome structure. And it turns out that condensin does something more than just make a nice helical staircase up the chromosome. So what we're going to do now is we're going to use SMC2, the SMC2A mutant, And we're going to compare what happens when a cell enters mitosis with condensin and when a cell enters mitosis with no condensin. There were two condensin complexes, condensin 1 and condensin 2, but when SMC2 is in both of them, so if you have no SMC2, there's no condensin. And what I want you to see now is what happens when we release these cells into mitosis. 5 minutes. In wild type on the top, all the long-range interactions have melted away. On the bottom, it looks just the same as it did before. 10 minutes. The bottom still looks just the same. In the absence of condensin, it still looks like an interphase nucleus. 15 minutes. Still looks like an interphase nucleus. So it looks like condensin blocks the cells from going into mitosis. At 45 minutes and at 75 minutes. By 75 minutes, the lower pattern is starting to become smeared out, but it still looks like an interphase nucleus. But if we look at what those, if we now look in the microscope at what we have, certainly by 45 minutes and by 75 minutes, if you were a cell biologist who worked on mitosis, you would say, oh, those are mitotic cells. But according to high C, they were all interphase. So what this says is that in addition to doing its job making a helix and condensing the chromosome, condensin has another job, and we don't yet know how it does it, but it must be involved in taking the interphase nuclear architecture apart. And condensin is an ATPase, and it may well be that condensin has to use ATP to take the nuclear architecture apart and form this new structure in mitosis. Now, just in closing, I want to say, what do the two different condensin molecules do? So condensin 2 was the red one that was making the helix, and in fact, if you redo these sets of experiments now and just knock out condensin 2 by knocking out one of its specific subunits, if you have no condensin 2, you see the diagonal line, but in the lower sample at 30 minutes, there's no paired lines on either side. There's no helix. So no condensin 2, no helix. With no condensin 1, there is a helix. But actually, you can see that the two outside lines on the bottom right are further apart and bigger and fuzzier than the two on the left. So in fact, with condensin 2, you have loops, you have a helix, but you have much bigger loops. So this is a summary of that. In wild type, you have nested loops inside one another with condensin 2 making a helix that has 400 kb loops, and condensin 1 making 80 kb loops inside that. If you have no condensin 2, it's not helical, you have 80 kb loops. If you have no condensin 1, it is still helical, but the loops are far too big and the chromosomes are too large. So conclusions about mitotic chromosome structure. Mitotic chromosome condensation involves a reorganization of chromatin packing from this interphase organization of TADs and compartments to loops. The loops project outwards from a helical staircase-like scaffold. That scaffold is formed by non-histone proteins, by condensin 2, in fact. The condensin 2 complex is a key complex that assembles the scaffold on the inside, and condensin 1 associates with the periphery and helps to further condense the chromatin. And condensin, in addition to doing this work on building the chromosomes, is also required to take the organization of the interphase nucleus apart as cells enter mitosis. So you end up with a structure like this, where there's a helix up the middle of the axis, which is the red loops. The loops are very, very densely packed next to one another, so that if you look at the bottom of all of this, it looks like random spaghetti. But in fact, it's a helical organization with loops, as I have told you. So I showed you some old electron micrographs that I did. I showed you some proteomics that Shinya Ota and Yuri Rapsilber did. All of the cell biology and the work which is coming out in Science was done by Kamiko Samajima, who is a fabulously talented senior postdoctoral associate. The 3D, the electron microscopy was done by Dan Booth. The high C was done by Johan Gibkus and Job Decker. And the mathematical modeling was done by Anton Golub-Dorodko and Leonid Mirny's lab. And this was the most, I've been involved in many collaborations in my life, and this was the most fun collaboration of all of them. And nobody could have done anything without the other people. And here's a typical day at the office for the team, because since we weren't in the same continent, we met by Skype continuously, and we had a great interaction. So thank you for listening to me, and I'd like to thank the Wellcome Trust for supporting the work in my lab.